Whenever we do a gut extraction, it's always a good idea to check the status of the gut contents. So we do that by dissecting a termite and just making sure that it's representative of things that we've seen in the past. We'll actually do that visually by looking at the cells in this microbial community. Just at the very tip of the abdomen here. And with one pull, you should see the gut tract. So this is just a buffered salt solution that matches very closely the composition of the actual gut fluid of the termite. And I'm just going to macerate this up a little bit, and then I'll add it to the microscope slide and we'll have a look. Okay, and we're ready to look at that under the microscope. Yeah, that looks really good. Termite gut, there are representatives of all three recognized domains of life. There are eukaryotic protozoa, there are archaeal methanogenic organisms, and then there are also bacteria. And our lab is predominantly concerned with the bacterial population in this environment, namely the spirochetes. In this particular type of termite, spirochetes are among the most dominant of the bacterial members of the community. And our current research shows that, in fact, they do engage to a large extent in CO2 reductive acetogenesis. So these are just various different types of protozoa, and they're thought to participate namely in the primary breakdown of wood. So they're thought to contain xylenases and cellulases that allow their enzymes access to these wood polymers, and then they ferment those into sugar monomers and polymers that are further broken down by other members of the community. And these protozoa also produce acetate themselves, but one of their byproducts is hydrogen and CO2. And this hydrogen and CO2, again, is what's used or consumed by the acetogenic populations in this environment. And here you can just see a swarm of protozoa organisms, probably right next to a very large species of trichonympha there. It's one of the largest organisms in the termite gut. As you go down and you actually look at areas that don't contain these large protozoal cells, you'll see that the background, this sort of interstitial fluid, is also loaded with spirochetes and other bacterial cells. And so, the complexity doesn't get any less even in those spaces. One of the things that I should point out is that the typical volume of the hindgut paunch, such as the one that we looked at here, is anywhere from 2 to 5 microliters depending on the size of the termite. And so the wet mount prep that I've made here really represents at least a 1 to 20 dilution of that environment. And so it's actually, these species are packed in much more densely in the actual intact gut than what's represented here. So that gives you some idea of the density as well as the diversity of life in this